Hello, you're welcome in this session. And today we want to talk about meiosis, which is type of cell division. We have completed mitosis, which occurs in the somatic cells and even the reproductive cells. So we are going to look at now meiosis, which specifically occur in reproductive cells. So meiosis, for it is restricted into the germs. Those are the reproductive cells, that is the sperms and the oocyte. That is where meiosis occurs. And this meiosis we are talking about, it is a type of cell division where the original cell, if we have the original cell, it divides to give two daughter cells. It gives two daughter cells, and these two daughter cells have half number. They have half number of chromosomes as the parent. So we saw in mitosis they were dividing to give the identity, they were identical, having the same number of chromosomes as the original parent, but within the somatic cells, we see them undergoing meiosis to form haploid cells. So these cells which we are seeing here, they are called haploid. So we have 2N, and we are separating into N and N. So we produce haploid cells. And this one, its significance is during fertilization, we see the sperm joining, the nucleus of the sperm joining together with the nucleus of the egg to form a zygote. So if these cells do not form half number of chromosomes as the original cell, during fertilization to form a zygote, they can double. So to prevent that, we break them down into half number as the chromosome, that during formation of the zygote after fertilization, they again form the same number of chromosomes as the original parent. That is its major significance of meiosis. And this meiosis we are talking about in this session, it occurs in two stages, whereby if we have meiosis, it undergoes what we call meiosis 1 and meiosis 2. And this is the one that is different from mitosis. We shall see it differing from mitosis majorly in prophase. But other stages from metaphase, anaphase, and prophase is the same as mitosis. And even meiosis 2 is purely the same as mitosis. It's the same. So the difference here, we are seeing it in prophase 1, not that one. That during meiosis, the only difference between it and the mitosis is that they differ in the prophase 1 of meiosis 1. Other stages are the same as mitosis. And that one makes our life simple. After we have known that all the stages are the same as mitosis, and we have already known mitosis, then we can apply that knowledge for the studies. So the major thing here to understand is prophase 1, where I want you to pay attention to see the difference between meiosis and mitosis. So in meiosis 1, we are beginning with meiosis 1, and this meiosis 1, specifically we are beginning with the prophase 1, which is only different. And in the prophase 1, this prophase 1 is divided into sub-5 divisions, whereby we have liptotin, we have zygotin, we have pachetin stage, we have diprotein, and lastly we shall talk about diakinesis. And these are the 5 stages that occur during prophase 1 of meiosis 1. And we have a mnemonic to always remember, and this mnemonic states that let Zanzibar and Pemba demand democracy. So I can always use this mnemonic to remember the 5 subdivisions of prophase 1, whereby Let is for liptotin, then Zanzibar is for zygotin, Pemba is for pachetin, then demand is diprotein, and democracy is for diakinesis. So that is how I can always remember the 5 subdivisions of prophase 1 by saying Let, Zanzibar, and Pemba, demand democracy. So after knowing the different stages, we want to see what happens under each stage. And during liptotin, lipto means thread-like or thin. So meaning during liptotin stage of meiosis 1, this is liptotin, liptotin stage. We are saying the cell, in the cell, the chromosome is thread-like, or it is thin. So it is formed up of thin thread-like chromosome, and these chromosomes, they are long and thread-like. So that is majorly what we see under liptotin, meaning thread-like. So the chromosomes are still thin and not visible very well with our eyes. So we cannot see them visibly, and they are thin, but they have the centromere, whereby they are still thread-like. They haven't thickened to form bigger chromosomes. So under liptotin stage, what you mention is that you tell us that the chromatin are thin, the chromosome is long and thin. So that is the major thing we see under liptotin stage. And after liptotin stage, it drives us into zygotin, and under zygotin stage, zygo is a Greek word which means pairing. This one is the same as pairing. That is what zygotin means. So under zygotin, we are going to see the sister chromosomes. This is the chromosome, this is the first chromosome, this is the second chromosome. So the sister chromosomes pair up. They pair up. And after pairing up, meaning they come closer to each other. If this is the chromosome and this is another homologous chromosome, they pair up and they come together near each other. And that is what we are calling under zygotin. And this process whereby the two sister chromosomes, the two homologous chromosomes pair up together is what we call this one synapsis. This process of pairing up is known as synapsis. So synapsis is the pairing up of homologous chromosomes. They come near each other. That is what we call. So the sister chromosomes the homologous chromosomes having the sister chromosomes, they pair up near each other, and by the process known as synapsis. That is during zygotin, whereby zygos means pairing. And that was the second. And the third stage is pachetin stage. And pache means thickening and crossing over. So we are going to see the homologous chromosomes which are in this cell. They are still these homologous chromosomes in this cell. They are going to cross over. So if I have a chromosome here, this is a chromosome, this one like this. So I'm going to form another one. This other one, find it crosses over. So the importance of pairing up is to enhance crossing over. So during the pachetin stage, we see the cells crossing over, and at the point where the cells cross over to share the genetic material. So here they cross over and start sharing material. At this point where they meet is what we call chiasmata. So they meet at a place known as chiasma when they are many, and when it is one, it's called chiasmata. And this process of coming together, they form a tetrad of a four chromatin. The non-sister chromatids cross over at chiasmata to form a tetrad, and this process is what we are calling crossing over. Crossing over. Crossing over of non-sister chromatids. Non-sister chromatids. What do we mean by non-sister? When this is this, these are sister chromatids, but if this one crosses over with this, these are non-sister. They are not homologous. But this and this, they are homologous. So that is what we call, and crossing over, the site where they cross over is called chiasmata. And this is during the Pachetine stage. And from Pachetine, which is number 3, we go to the step number 4, which is diprotein. Diprotein stage, whereby we are still using a mnemonic Zanzibar and Pemba demand democracy. And in this process, we are going to be talking about the diprotein stage of metaphase 1, of prophase 1 of meiosis 1. And dipro or di means two or separate. So we are going to see, as much as these homologous chromosomes have crossed over using non-sister chromatids at the chiasmata, we are going to see them moving apart. So the homologous chromosomes during the protein, they move apart. They move apart, but as they move apart, they remain attached together at the chiasmata. Remain attached together at chiasmata or at the chiasma. The chiasmata. So they move apart. So di means they are moving. But when they are moving, they are still attached together. And in that movement, they are sharing genetic material. So it is during this movement where they share genetic material. So that is during the protein stage. And finally is the diakinesis, which is number 5, which we are calling diakinesis. So here they are going to move in different directions. Here the cells move in the chromosomes, they move and pull apart and they separate. So we are going to see these chromosomes which were attached at the chiasmata. They, after exchanging the genetic material during the protein stage, they pull apart. They separate completely. Here they were separating but not completely. They are separating but still attached at the chiasmata. But here they move apart and align, as the way we are going to see it under metaphase, aligning at the equator of spin. So the homologous chromosomes here, they separate. So here it was still a tetrad, and even at the protein stage, it was still forming a tetrad. The tetra means 4, but here they have pulled apart and they are no longer joined. The sister chromosomes, the non-sister chromosomes are no longer attached together. So under diakinesis is complete separation. Complete separation of the non-sister chromatids from the chiasmata. So that now they are free together to start separating to form haploid cells. So these are the stages under prophase, and other processes which are remaining are the same as mitosis. So we have seen the differences in the prophase 1, whereby we have 5 subdivisions, that is late Zanzibar and Pemba demand democracy. That is liptotin stage, zygotin, then pachetine, diprotein, and diakinesis. And after they have moved apart, we're going to see them during metaphase. So this was prophase 1, now this is metaphase 1. So during metaphase 1, we are going to see the chromosomes now after exchanging the material and after crossing over, having now this crossing over is the one that brings about genetic recombination. Genetic recombination, that is the formation of new combinations in genes, and it is the one that is responsible for bringing variation in a population, whereby some people are short, others are tall, others are brown and black. It's brought about by crossing over, and this crossing over which occurs during the Pachetine stage of prophase 1. So in metaphase 1, we see the cells align at the metaphase plate the way we saw it in mitosis. They align at the metaphase plate, or you can call the equator of spindle. After aligning, we go to anaphase 1, and in anaphase 1, we're going to see the pulling apart. They pull apart to form a V shape. They pull apart the sister chromatins, and with this pulling apart, these chromatins which are being pulled apart, the chromatids, they have half number of chromosome as the original parent. And remember, it is during late anaphase that we form a furrow. That is where cytokinesis begins from. And then we proceed into telophase. And in the telophase stage, we are going to see complete cleavage of the cell after the nuclear envelope reappearing, after the nucleolus reappearing, whereby these guys have reached the opposite poles. Now even the nuclear material become thread-like. They become thread-like. The chromosomes become thread-like. Then the nucleolus reappear, the nuclear envelope reappear. And then we cleave, that is cytokinesis, producing two daughter cells. But these two daughter cells we are producing for them, they have half number of chromosome as the original parent. So they are haploid. We are producing two haploid daughter cells. That is, it occurs during meiosis 1. So after producing two haploid with the nucleolus, the nucleum, the chromatin, or the thread-like chromosome with the nucleolus, after producing these two daughter cells, having half number of the chromosome as the original cell, this is the end of meiosis 1. And after forming these two cells, we're going to see each of them undergoing mitosis. We shall see this haploid cell, it will also undergo mitosis. That is, it will undergo meiosis 2. And also this one will at the same time undergo meiosis 2. And I told you meiosis 2 is exactly the same as mitosis. So we are going to see this one undergoing still PMAT, prophase, metaphase, anaphase, and telophase. It will also bring 2. So at the end of this, it will also produce other 2 cells which are identical to it, which are, they also have the half number of chromosomes as the original, as this. For them, they will remain with the half number. So they are identical to this, but this one is not identical to this. We shall see even this one undergoing meiosis 2. At the end, it will also produce other 2 cells with half number of chromosomes as the original parent. And at the end of meiosis, we had one cell, original cell, but at the end we have formed one cell, second, third, and fourth. So at the end of meiosis, we have one cell producing 4 cells, each having half number of chromosomes as the original parent. This is what we can discuss under meiosis. So in summary, I have told you that meiosis occurs in reproductive germ cells, that is the sperms, and in the oocyte, or what we call the ovum. And we have seen they undergo what we call, or they produce daughter cells. And actually the end product of meiosis is the 4 daughter cells, each having half number of chromosomes as the original parent. So they are haploid. That's what we say, they are haploid. And we have seen meiosis undergoes two stages, that is meiosis 1 and 2. And we have seen in meiosis 1, it is the same except in prophase 1 of meiosis 1, whereby prophase 1 is subdivided into 5 stages, that is liptotin, zygotin, pachetine, diprotein, and diakinesis, which we have used the mnemonic Leti, Zanzibar, and Pemba, demand democracy. And we have seen what happens in each stage, whereby we have seen in liptotin, lipto means they are thin and thread-like chromosomes. And then we said in zygotin, zygos means pairing up of the sister or the homologous chromosomes. They pair up or they come together and they form by the process known as synapsis. That is what happens there. And the pachetine, we have said, it is crossing over. They shorten, thicken, and then the non-sister chromosomes cross over to the chiasmata, and they form what we call the tetrad, which is 4, means 4. They form a tetrad. And it is this crossing over during pachetine that brings about genetic variation or recombination or formation of new genes in microorganisms. And we have seen also diprotein, which is separation, pulling apart of the chromosome, homologous chromosomes, but still attached at the chiasmata. And it is during that process of pulling apart that the genetic material is shared at the chiasmata. And then finally is diakinesis, whereby they completely separate to prepare for metaphase. Then from metaphase 1, anaphase 1, telophase 1, and then after telophase 1 produces 2 daughter cells with half number of chromosomes as the parent, and each cell undergoes meiosis 2, which is the same as mitosis. This one produces 2 cells, and also this produces 2 cells. At the end, we have formed 4 daughter cells. And the significance of this, we told you, it is during reproduction we see the sperm cell and the egg joining together to form a zygote. So we are forming half number of chromosomes that during fertilization they again form the same number of chromosomes. Thank you so much for listening till the end. Remember to subscribe and share your comments and like the video. Thank you so much for listening.